Hello, welcome to the second video, which is all about endogenous pacemakers and exogenous zeitgebers. As we know that there is various features which we need to cover for this video, and they would include us looking at the SCN, or the suprachiasmatic nucleus, and the various exogenous zeitgebers alongside all of the case study material. So we've got the internal, the biological clocks, which are known as the endogenous pacemakers, and then we've got the external environmental cues known as the exogenous zeitgebers. Now, these have all maintained and played major influence on our lives, and throughout this essay we are going to discuss this further. In the lesson we learnt about four main biological rhythms, and they are called the circadian rhythm, the ultradian rhythm, the infradian rhythm, and the extra rhythm, which we've learnt about, is the diurnal rhythm. Now, the circadian rhythm is one of the most dominant rhythms within our body. This is roughly about every 24 hours and is associated with the sleep-wake cycle. A subsection of the circadian rhythm which we know about now is the diurnal rhythm, which is controlled by day and night. And as we learn about endogenous pacemakers and exogenous zeitgebers, this rhythm has got a lot of interaction with what we are learning about. Now, the ultradian rhythm, this happens less than once a day, and it's shorter than a day. An example of this would be our heartbeat. We've got the infradian rhythm, which happens more than once a year. An example of this is the menstrual cycle. All are controlled by both internal and external forces. And as we discussed throughout the whole of this essay, this needs to be a focus. Now, the main bit of modern research that we will be including in this essay is the Jessica Campbell talk from TED Talks. Now, the main portion that we are pulling from her whole dialogue, her whole speech, is the fact that she spoke about the horseshoe crab. Now, the horseshoe crab, as she outlined, its rhythm is not as malleable as what a human's rhythm is. If they are put in a new location, they will continue to have the same rhythm that they would have in their home space, and they'd maintain this within their new location. Obviously, for us, when we are looking and discussing about rhythms, we identify and we talk about the whole fact that human rhythms can change, etc. Whilst actually, when we're looking at research that includes animals, it's highly problematic that we include and take into account animal research, because as we should know by now, the rhythms, even though they look the same, however, they may not have the same type of changeability that the human rhythms have. Now, the main endogenous pacemaker that we look at is called the suprachiasmatic nucleus. Now, this is a tiny cluster of nerves just located above the optic chasm, and it controls the amount of melatonin that is produced within before you get to sleep. And then sends signal to the pineal gland, which then, if there is lots of light, it tells the pineal gland to stop producing the sleep hormone called melatonin. When a decrease in light is detected, the pineal gland stimulates production of melatonin, and melatonin then induces sleep as a response. The SCN, therefore, is one of the main endogenous pacemakers that controls the sleep-wake cycle throughout the day. As we can see here, that the SCN is going to be focusing down and controlling the circadian rhythm. However, as we have learnt, the diurnal rhythm is directly controlled by the SCN as a result, due to its interaction with day and light. Now we're moving on to the section, or the AO1 section, about exogenous zeitgebers. So rhythms are set off by external stimuli or other features in the environment, such as light, temperature, and social cue, which are the main three exogenous zeitgebers that we'll be looking at. Now, they control and they interact with the endogenous pacemakers, such as the suprachiasmatic nucleus. Now, this is using cues from our environment, such as light with the sunrise and sunset. This then entrains our body to being in contact with the external world. Now, the three main exogenous zeitgebers which we're looking at, or time givers, are light. social cues, and temperature. Now, light can reset the suprachiasmatic nucleus, as we know through our dialogue interaction with it already, and it can also reset other oscillators, which are proteins that run through the body that are sensitive to light. So Campbell and Murphy found this. If you shine a light on the back of a participant's knees, the oscillators pick up the light and then they shift the biorhythms as a result. Now, social cues such as eating are appropriate for the age of the individual. So we all have various social cues that are determined by our lifestyles, etc. They found with mealtimes an interesting occurrence happened, which is the Zeitgeber for the cells in the liver are likely to be mealtime related as they are reset by eating. So the social cue of eating or mealtimes is what made those liver cells being produced and which are only reset by the consumption of food. Temperature, so this is obviously seen with the patterns of hibernation and the changing of the colours of the leaves. Temperature as a result resets their rhythms as to when it is appropriate as a result. And as we know, the suprachiasmatic nucleus relies on light, so we've got to take into account that with our evolving world there are going to be more features which interact with our lives than what they did in the past. So for an example, we have more artificial light in our lives, so we are exposed to more light in the evenings with artificial lighting, also interaction with our mobile phones, all of which can obviously impact our sleeping habits negatively. Changing lifestyles or cultures, this is also, individuals start to adopt a 24-hour culture where we may be asleep during the day and awake during the night as a result, depending on the type of job in which you've got. And obviously global warming, fluctuating temperatures can also have a negative impact on the environment as a result. Aschoff and Weaver here in 1976 conducted an experiment in a World War II bunker. These researchers placed participants in an underground World War II bunker and excluded any environmental or social time cues. This allowed their endogenous pacemakers to be free running. They found that most people displayed circadian rhythms of 24 to 25 hours, though some rhythms were as long as 29 hours. Now, what does this research say about endogenous pacemakers and exogenous zeitgebers? Let's look at this now. Okay, now what did Aschoff and Weaver show us about endogenous pacemakers? So without external cues, endogenous pacemakers were still able to maintain that 24-25-hour circadian rhythm, which means that potentially leaning towards the idea that endogenous pacemakers are the most important and most influential in maintaining our biological rhythms, such as our circadian rhythms. But for the case of exogenous zeitgebers, it does argue the case that possibly some individuals do need these exogenous zeitgebers to reset their rhythms, because a 29-hour rhythm, as one of their participants showed, would not be comfortable for the individual to have in today's modern society. So just leaning towards the fact that exogenous zeitgebers, they are important and they can be influential for some individuals' lives. Okay, now we're moving on to Morgan and his mutant hamsters. So Morgan bred genetically mutant hamsters which had circadian rhythms of 20 hours instead of 24 hours. He then transplanted the SENs, or the suprachiasmatic nucleuses, into normal hamsters. These normal hamsters then displayed the mutant rhythms of 20 hours. Now just take a few seconds to ponder, what does this research say about the SEN? Now let's look at this further on the next slide. What does this research say about the SEN? So the SEN, quite clearly from this research, it is the main endogenous pacemaker of the circadian rhythm or diurnal rhythm. Normal hamsters then start to display the mutant 20-hour cycle after the transplant happened, despite the presence of exogenous zeitgebers. So we've got to take onto the account that actually maybe the endogenous pacemakers are the main thing that control our bodily rhythms. However, we have got a few problems thrown into this research, especially because it is animal research. So we've got to take caution when extrapolating these results and applying them to humans. Jessica Gamble is a good person to bring in at this point, because Jessica Gamble really does highlight the idea that actually there is a difference between animal cycles and human cycles. Animal cycles are not as malleable as what human cycles are. The example of this should be taken from Morgan and his Mutant Hamsters, to the idea that maybe we shouldn't apply the findings of a nocturnal creature to a diurnal creature, which is what a human rhythm or human is. Okay, this is extension material, but I'll run through it just so then you've got this as revision material. So, Michelle Cifra, which is the cave study. So the aims of the study was to investigate what would happen to people's circadian rhythms if they were cut off from all zeitgebers and had to rely on their endogenous pacemakers to tell them when to eat and sleep. Now, Cifra was a French cave explorer who spent over 6 months living in a cave in Texas, deep under the ground, with no natural light or anything else to tell him what time of day it was. His biological clock was allowed to free run. That is, he just allowed the body's inclinations, eating and sleeping whenever he chose, with no fixed timetable. He was wired up so that some of the body functions could be recorded. He had a telephone linked to the outside world and was monitored by video camera. It's important to note that although the circadian clock could be reset using artificial light, Cifra had no way of telling time, and his use of artificial light did not help him maintain regular sleep-wake cycles. So the results showed that he had fairly erratic sleep-wake pattern at first, but it settled down to a sleep-wake pattern that averaged out to be over 25 hours instead of 24 hours. Now, what does this research say about EPs and EZs? Let's look at this on the next slide. The most notable thing that we draw from the study is that the endogenous pacemakers are more efficient in maintaining the circadian rhythm. When allowed to free run, his sleep-wake cycle stayed around average 24 to 25 hours. Perhaps exogenous zeitgebers are just not as important as we once first thought. This also starts to bring in the idea that maybe the body's natural circadian rhythm is about 25 hours when it's left to free run. Jessica Gamble, in her TED talk, also tells us about this, as the shift of time and culture, etc., did lead us to creating this 24-hour culture when our bodies are naturally used to this 25-hour rhythm. Another issue with this research is it is a case study, so it's a unique thing that's happened. So we've got to draw into question here, is it generalizable? Can we really start to apply this to everyone when it may just be a unique thing to that individual? Next case study, which is Campbell and Murphy, which is the light and the knees experiments, is relating to exogenous zeitgebers. Now, Campbell and Murphy in 1998, in a bizarre experiment, shone bright lights on the back of participants' knees and were able to alter their circadian rhythms in line with the light exposure. The exact mechanism for this is unclear, but it seems possible that the blood chemistry was altered, perhaps by the detection from the oscillators. Remember, they are proteins that run through the body, and this was then detected by the suprachiasmatic nucleus. Now, what does this research say about EPs and EZs? What does this research say about EPs and EZs? Now, the most thing that it's highlighting here is that eyes are not the main things that are used to detect light. There are proteins called oscillators throughout the body that can detect light and can potentially shift our rhythms as well. Now, what Campbell and Murphy are really highlighting here is the exogenous zeitgeber, such as artificial light, is just as perhaps as effective as natural light in resetting the suprachiasmatic nucleus. So we've got to start taking into account these new modern inclinations that are happening. Now, the next case study that we are moving on to is Miles et al. In 1977, the blind man case study. Now, Miles et al. In 1977 reported their case study of a blind man who had a daily rhythm of 24.9 hours. Other zeitgebers, such as clocks, radios, and social cues, etc., fail to reset the endogenous clock. This made it very difficult for him to function, and as a result, the man relied on stimulants in the mornings and sedatives at night in order to maintain a 24-hour sleep-wake cycle and get his biological rhythm in time with the rest of the world. Now, take a few seconds just to ponder these following questions. What does this research say about endogenous pacemakers and exogenous zeitgebers? And what particular exogenous zeitgeber can you comment on here? Okay, now let's look at what does this research say about EPs and EZs. Well, light is the most important exogenous zeitgeber for the circadian rhythm. However, without the detection of light, his circadian rhythm could not be entrained to 24-hour cycle. And this meant as well for us that other elements in his life, such as social cues, were just not enough to maintain that 24-hour rhythm for a day. So what did the blind man need to rely on? He needed to rely quite heavily on stimulants and sedatives to try keeping time with the daily rhythm of the world. Now, what does this mean for us? So this case study, if we are going to use Michel Cifra, is actually starting to support him because he found that he had an average of a 25-hour cycle. The blind man in this case study has an average of a 24.9-hour cycle. So quite possibly, when we leave our rhythms to free run, a 25-hour rhythm is the most natural. So this is showing support for Cifra. Whilst we're going to carry on with the criticism of this is a case study, so is it a unique thing? Can we really generalise this to the wider population? Start to draw this out within your essay. Not just natural light. Even though, yes, they are drawing us to attention that bright light was most effective, dim light was still possible and still could entrain our biological rhythms as a result. So, massive support for that exogenous zeitgeber as a result. However, the extension material for this, it raises implications for how we live in society. With so much artificial light around us and that we are being exposed to, this could be contributing to the increase in sleep and mood disorders because individuals are unable to maintain or have a good quality of sleep due to so much light being present in their environment. Okay, the next bit that we're moving on to is Davidson et al. In 2006, which is the mealtime study. Now, they found that the zeitgeber for cells in the liver is likely to be the social event of mealtimes because their rhythm is reset by eating. Now, what can we say about this study? Now, for us, this is highlighting the fact that light is not always the main exogenous zeitgeber for all cycles. For this particular cycle with the liver cells, social cues prove to be more important and more beneficial because they are able to entrain liver cells, whilst having something like light being entraining those liver cells wouldn't prove to be an adaptive mechanism, and we wouldn't be able to reset it as more efficiently as we do with the exogenous zeitgeber of social cues or meal times. Kelley et al. In 1999, which is exogenous zeitgeber AO2. They found that even with all external cues available in a submarine, lighting, social events, and temperature, this was not enough to shift the rhythm of melatonin secretion onto the artificial 18-hour day which was needed for their working rhythm. The rhythm instead remained at a 24-hour rhythm. Now, what does this research say about endogenous pacemakers and exogenous zeitgebers? What does this research say about EPs and EZs? As important as exogenous zeitgebers are, and we've proven with other case studies that we've looked at, and in particular light, these were not enough to override our endogenous pacemaker and change our circadian rhythm to an artificial 18-hour day. This suggests endogenous pacemakers are the most dominant here, and they are naturally maintaining our rhythm into that best 24-hour cycle that is needed to be an efficient member of society. This could also question the effectiveness of artificial light on melatonin, and because it wasn't enough to obviously reset and change our rhythms to an 18-hour day. Now, a few last points to raise here. Total isolation experiments seem to suggest two separate categories of endogenous or exogenous. However, such neat divisions do not seem to exist between the two. The question should not be which is most important, but how they work together, and this combined effort makes the most sense. E.g., the body needs an internal pacemaker, so processes are coordinated. Secondly, these rhythms then need to be fine-tuned by external cues so that we can keep in time with our changing world. Now, these symptoms that often arise, as Roth in 2007 raises here, can include irritability, insomnia, fatigue, etc. And these will obviously impact the individual's lifestyle in that new environment. Now, we have obviously got evidence for this, and I draw your attention to the baseball players who, as we could quite clearly see from that experiment, their winning streak started to decrease when they're experiencing the symptoms of jet lag. So it can have a negative impact on the individual when they are travelling or going through these different time paces without being synchronized with their external environment.